All right, so very good. Again, welcome everyone. This is our sixth chemistry seminar for this semester. And I have the pleasure of introducing Rika Isaac. And there's a little change up in how we are going to do this, at least in terms of the responsibility. So Rika will be our first official co-host, not just an introducer, but she'll be our co-host along with me. And so she will also help and participate in the question and answer period. So Rika is one of our biochemistry students. She is in her second year. In fact, she's conducting research now with Dr. Brian Wong over in the biology department. And her research involves breast cancer. And she's originally from Loma Linda, California. She went to high school at Loma Linda Academy. She plans to become a physician, but interestingly, she wants to specialize. She plans to specialize in neurology, to become a neurologist. And so there is some overlap there between today's presentation and neurology, I would assume. And she, for fun, I think, is it fun or is it something that are you in the band or something here at Andrews, Rika?
Yeah, I'm in the Wind Symphony.
Oh, you're in the Wind Symphony. Okay, so it's more than just fun. So she plays the flute here at Andrews in our Wind Symphony. So Rika, go ahead and introduce our speaker for us today. Thank you very much.
Thank you, Dr. Murray, for the introduction. Today, I have the honor of introducing Dr. Marchetto. Dr. Maria Marchetto is an assistant professor in the anthropology department at the University of California, San Diego. Dr. Marchetto earned a Bachelor of Science in Biological Sciences in 1998 and a PhD in microbiology in 2005 from the University of Sao Paulo, Brazil. She moved to San Diego in 2005 for her postdoctoral research in stem cells and neuroscience in the lab of Dr. Fred Gage at the Salk Institute. Before her appointment at UC San Diego, Dr. Marchetto was a staff scientist at the Laboratory of Genetics at the Salk Institute, where she led projects that studied neuronal development using human and non-human primate stem cells. Today, Dr. Marchetto will be presenting on the molecular mechanisms of bipolar disorder. Her current work focuses on using patient-derived induced pluripotent stem cells to study the cellular behavior of human neurons in neuropsychiatric and neurodevelopmental conditions, such as autism and bipolar disorder. In addition to her work on bipolar disorder, Dr. Marchetto's work is also focused on human evolution in the context of human brain expansion and neuronal development. Her innovative approach uses stem cell models from human and non-human primate species to test hypotheses about the role of disruptions to human-specific cellular and molecular signatures in development and their potential impact on mental health. Please join me in welcoming Dr. Marchetto.
Thank you so much for the kind introduction, and thank you, Desmond and Andrews University for inviting me. Happy to be here. Today, I'm going to talk about some of the work I did back at the Salk Institute and in collaboration with my lab now at UCSD. And we study, the idea of this project is to study molecular mechanisms that could be cellular and molecular mechanisms that could be involved in the pathology, the brain pathology that happens in bipolar condition, and understanding the underlying mechanisms, cellular mechanisms of these conditions can help potentially create new therapeutic approaches. So that's the idea. So then I'm going to show you one of the aspects and we'll illustrate how do we study cellular and molecular changes in the lab using cells from patients with or individuals with bipolar disorder. And I will hopefully reveal to you that there is a molecular pathway that involves beta-catenin, and one of the components is a gene protein that expresses a protein called LEF1, and that expression is I'll hopefully show you that is associated with lithium resistance and hyperexcitability in neurons derived from bipolar patients. So that's a mouthful, but hopefully we will walk together that path. And please feel free to ask me questions. I can't access the chat and the talk at the same time, so I will maybe look at the chat after finishing, or maybe if you have a very pressing question on something I said that is hard to keep following, you please you can be free to interrupt me.
Yeah, we will monitor the chat for you.
Thank you.
Okay.
Okay. Advance my slide. So bipolar disorders is a umbrella of conditions where the individuals experience mania and hypomania, as well as moments of periods of depression. And there are many specific criteria that clinicians can use to identify an individual with mania and hypomania, followed by depression. And those can help a clinician identify the individual as a bipolar patient or a major depression, a patient with major depression. Those are very two different things, and lines of treatment and approaches are very different too. So the population risk is between 1% and 2%. There is a level of heritability between 80 and 85%. Monozygotic twins concordance is between 40 and 45%. That means that if you have an identical twin and you experience bipolar condition, that is up to 40. Identical twin sibling will also have developed symptoms of bipolar. And there are some, and given that heritability and that concordance between monozygotic twins, there have been many studies showing that there is a strong genetic component. And some of those studies are genome-wide association studies showing that there are some genes, and I'm giving some examples. An example will be CACNAC1, which is an alpha subunity of a voltage-gated calcium channel, have been involved with bipolar, enriched in bipolar patients, even though mutations in that gene, even though there is not a specific gene that is directly responsible for bipolar. So what we believe now, and also genetic linkage has shown other genes that are involved in different types of molecular pathways. I'm citing here GSK3, which is a glycogen synthase kinase. It's a kinase, genes involved in glutamate transmission, stress response, inflammation, apoptosis, and oligodendrocyte-mediated myelination have been involved in or associated with bipolar. It doesn't mean that those genes will cause, mutation in those genes will cause bipolar, but those genes, mutation in those genes have been associated with, found more often in individuals with bipolar disorder. That is important to us in the bench work, because if we want to be able to isolate cells from a patient that has bipolar, completely away from that person's environment and culture those cells in a dish, it is important to trust that there will be a genetic component too, and that will make it the disease modeling in the dish more better and more truthful, because we'll be able to reproduce some of the genetic changes, because if we get a patient that have a potential mutation in some of those genes, we'll be able to hopefully see changes in the dish. So bipolar first line of treatment drugs is a mood stabilizer. Those are medications that can decrease vulnerability to subsequent episodes of mania or depression. So it keeps the mood stable and do not exacerbate the current episode that the individuals are going through. The most well-known and most used mood stabilizer is called lithium. It's a salt, and it's given as a lithium carbonate. And the salt is given as a mood stabilizer drug, and that's usually the first line of drug given. The caveat is between 30 to 40% of bipolar patients don't respond well or don't tolerate lithium treatment. So when you look at a patient population, you're leaving 30 to 40% of these patients without one of the most efficient treatments. So we had this idea of approaching that question in the lab from a drug response perspective. So we took cells from neurotypical controls, which are individuals that don't have any, at least they don't have a diagnosis of any known neurological or neuropsychiatric condition. And we took patients, sample cells from patients with bipolar that were either responsive to lithium treatment or non-responsive to lithium treatment. And those are patients that came to us, the cells from them came to us after, in collaboration with very skilled clinicians, John Kelso at UCSD, Martin Alda at Dalhousie, Canada. So the idea, I'm going to tell you what we have done with these lines. We took the somatic cells, the cells that we can easily access from the body, which are fibroblasts or lymphoblasts. We differentiate, we reprogram them. So we change the program of a fibroblast, of a lymphoblast, into an iPSC, which is an induced pluripotent stem cell. So those cells are now able to become in theory, all cells in the human body, but we are studying conditions that affect the brain. So we wanted to look at brain cells. So we differentiated using a number of factors that we have been established over the years. We are able to differentiate those induced pluripotent stem cells into neurons. So those are brain cells. And then the idea after lithium treatment is that if we see changes in those neurons from bipolar patients that are different from neurotypical controls, the idea is that only the patients that are responsive to lithium will respond to lithium in the dish. And one thing that we have noticed is that here shown on the bottom, both by calcium imaging and by electrophysiology, if you look at the right, two right panels, the cells from patients with bipolar, and here on the very bottom right corner, have more activity. And when you add the drug, the lithium, the activity is calmed down and decreased to levels of the neurotypical. So that's what lithium treatment does in the culture to neurons from patients that are responder to lithium. when the patients are not responding to lithium, we don't expect to see that response. So the cells are the neurons that are, we call them hyperactive. In bipolar condition, they will continue to be hyperactive regardless of lithium treatment. So that was the hypothesis that we started this project in 2015, many years ago, was that if a patient is responding to lithium, the cells will also respond to the drug treatment. And please don't read this whole slide. The idea is just to give you also a caveat that lithium have many downstream effects. And one of the effects is that it does inhibit a kinase called GSK3 beta, and consequent also decreases the induction of the canonical Wnt pathway. And that's one of the most studied effects of lithium, just so we understand a little bit, people do understand a little bit about the mechanisms in which lithium acts. Just so we can have a visual for that, here is the Wnt signaling pathway, and lithium can act by blocking beta-catenin, blocking GSK3 beta inhibition of beta-catenin. So as a consequence, when you have lithium in the system, there is an increase in beta-catenin activity. And beta-catenin is a transcription regulator that acts downstream of beta-catenin. There is many genes that can be expressed and regulated. So the idea here, without too many details, is the fact that bipolar patients respond to lithium was already telling us, respond or non respond, was telling us that beta-catenin pathway might be involved. So just to show you some of the previous data from the lab that showed that response to lithium in terms of hyperexcitability in cells from bipolar patients. We here in the left corner, I'm showing you again spikes. So those are the in blue from a control patient, and in red from a bipolar patient. And that's showing you that bipolar cells have many more activity, or much more activity, or many more spikes. And we can detect those cells in culture by using a reporter that tells us where the neurons are. So when the electrophysiologist is gonna do a patch clamp, which allows the electrophysiology to detect the levels of activity in a neuron, the person uses this indicator, which makes the cells glow green. So the summary of the activity is here plotted as total evoked action potentials. So you can, as you can see in red, bipolar patients have much more, cells have many more action potentials compared to controls. And that is also true. And then when you treat the cells with lithium, and here on the bottom of this page, we're calling LR lithium responder patients and NR non-responders. And when you look at the lithium treatment, minus is not treated, plus is treated. So treatment with lithium on lithium responder patients decreases the activity to levels that are comparable to control, and treatment with lithium on non-responder patients, or NR, doesn't change the activity profiles. So that is to show that lithium responder individuals are still responding at the cellular level to lithium treatment, and non-responders are not responding. Then we got another cohort of patients because we really wanted to confirm that that was real. And we used a cell line from different cohorts, and a different person was in the lab at that time, and a different clinician selected those patients. But again, the same cohort was responding and non-responding to lithium. And what you see here on the averages is for this different cohort, the same thing happens again. You see the lithium responders in red. After you add lithium, there is a decrease in excitability. Here shown the plots in B, without lithium on the top, with lithium on the bottom. And on the I and J, the right panels, you see the non-responders, the NR, where treatment with lithium doesn't change the hyperactivity profile. So this work was able to replicate the hyperexcitability phenotype in lithium and the lithium responsiveness in bipolar, and extended the previous findings. And we, for this work, Shani, which was the leader, also characterized the type of activity that was changing in those neurons, which it's a little bit too much to go into in the interest of time for this talk. So I'm going to move forward to the work that we did last year. We saw all these changes in activity. And then we wanted to ask the question, can we identify molecular changes that characterize lithium resistance in bipolar? If we can understand what could be underlying the inability of the patients to respond to lithium, could be potentially useful in moving forward with new therapeutic approaches. So we, to get some idea of what were, unbiasedly get an idea of what are the molecular pathways that were changing, what we did is we took an approach of looking over differentiation time and which genes were being expressed at any given time during differentiation. So we start those, as I mentioned before, we started with the induced pluripotent stem cells, and then we make neuroprogenitor cells, and then we make the neurons. So what I didn't tell you, I'm telling you right now, is that between the IPS and the neurons, there are many steps. And one of the steps is the neuroprogenitor step, which those are baby neurons. They're still able to divide, and they are able to differentiate into a more mature neuronal population. And for that, we use a time and a different number of exposure to different factors that will take a baby neuron, a neuroprogenitor cell, into different stages up until the more mature neuron that has all those phenotypes that we saw before in terms of hyperexcitability. So to understand what was going on over time with the different populations, the bipolar patient responders, non-responders, and the controls, we took RNA, which is looking at what all the genes that are being expressed in a given time. So that gives us an idea of the cellular state, and we sequence whole RNA, everything that the cell was making at different time points. So we started at neuroprogenitor cells, or we call it time 0, and then we look at 1 week, 2 weeks, 4 weeks, and 6 weeks old neurons. And that's the later, that's the as mature as we went in terms of neuronal differentiation. By 6 weeks, those neurons are firing and active, but some of them started being active earlier too. But we wanted to compare. So what we did is to have an idea of how this population looked. We plotted, if I can move, we plotted on a graph we call principal component. And we look at two principal components. So every dot here is it's all the cells is one cell line projected at a time. So in zero here in red, we have all the neuroprogenitors from bipolar patients, lithium responders, and lithium non-responders. And what we noticed when we looked at all the samples is that there was a clear distribution on PC1, which is the axis of the time, that we could see that the neuroprogenitors were distributed in a specific place. They were clustered together, and as soon as the neuron starts differentiating, there is a movement in the graph showing that the cells are clustering together. This was important for us to know that our neuroprogenitor cells were still neuroprogenitors. They might have differences, but they are not becoming something else. They are still progenitors, and the same for the more differentiated neurons. So pre-subcomponent analysis show the neurons are segregating by time point, which is important to know that even though they are bipolar neurons or control neurons or responder or non-responders, they are still neurons and they behave similarly. The other thing we noticed was, and that's just to confirm that the neurons from controls bipolar patients and responders and non-responders are still in general behaving similarly. Here, I'm just showing images on the top two rows are from neuroprogenitor cells. So those are the baby neurons. They do express genes that are related to maintenance of neuroprogenitor state. So those are highlighted here on the left, PAX6, nesting, and SOX2. And also at the neuronal level, they also express cells, genes that are expected to be expressed at those moments, to confirming that the percentage of progenitor markers and neuronal markers is comparable. So we're still making neurons. So that's important. And then we did a little bit more analysis showing other neurons and other markers using the RNA sequencing analysis to show that what we were making were actually neurons. So let's just cut, but it's okay. So double cortin, RBFOX3, DCX, and GFAP. So those are all markers for differentiating cells. So our next question was looking back at that principal component analysis. Remember, so those are exactly the same points, but now instead of plotting time or coloring by time, we're coloring by disease status. So are those controls, are those responders, or they are non-responders? And what we see is when we do that, we clearly see that the non-responders are seem to be separating from the controls and the lithium responders. It's almost like the lithium non-responders are a whole different thing altogether. We thought this was really interesting, and that's a principal component analysis too. So that's just looking at that axis. And the other thing we noticed is that divergence from controls and non-responders happens early during differentiation, right? So the divergence is already happening at earlier times. So we wanted to follow up on that. And then we did the second RNA sequencing analysis of bipolar and controls at a 2-week-old sorted neurons. So we have ways in which we can select just a neuronal population, and we use the FACS sorting for that. And we wanted to look at what those neurons were making, what these neurons are making comparable to controls and bipolar. And when we do that, we do notice that in general, bipolar neurons are different from controls with a certain amount of genes. But more interestingly, when you put them all together and compare lithium responders and non-responders with controls, we see that the number of genes that non-responders have that are different from controls that the non-responders are expressing, it's almost 10 times increased compared to the number of genes that lithium responders have. So that is telling us that non-responder is very different from both control and lithium responders. So what are those differences? So we wanted to look at the differences from non-responders when compared to control, because they were clearly many more than when you look at responders, right? So when we do that, we see changes in synapse genes, neuroprojection, DNA replication, calcium dependent, and we see changes in Wnt signaling pathway. Remember, I was talking about beta-catenin and Wnt signaling. So those are all in the same pathway. So Wnt signaling, that indicated to us that this signaling pathways may be involved in this unique profile that non-responder patient cells have. And when we look in more details, which are, so those are all the genes listed here in those two plots. They are all the genes that are related to Wnt signaling pathways. And when we look at some of the changes here in the values that are different in black, there are many more genes that are involved in the Wnt signaling pathway. They are different and significantly changing in non-responders than if you look at the lithium responders. So that's why it's a lot of gray bars. So those are not non-significant. But more interestingly, in the top of the list is this gene called LEF1. And here is just showing the plot for some of the genes that are differentially regulated. But I just wanted to focus on LEF1, which is very decreased expression, has very decreased expression in non-responders, very significantly decreased expression in non-responders compared to responders and controls. So with that, we did conclude that Wnt signaling pathway was differentially expressed in non-responders. Now, that's a little snippet of the Wnt signaling pathway for us to have an idea of a visualization. No need to spend too much time on the details and the names, but just wanted to show to you that indicated in blue and orange are all the genes that are part of the pathway that are differentially expressed in non-responders compared to control. So that's just to give you a visual of the amount of genes that are differentially regulated, right? And interestingly, I'm just pointing here some of the most significantly differentially regulated genes. One is the LEF1, and LEF1 is downregulated. Another one is responding to, which is the activator of the pathway. This one is also downregulated. And Frizzle-B is an antagonist of an inhibitor of the Wnt signaling pathway, and this one is upregulated. So, given those different upregulations and downregulations, it seems like, and we hypothesized that Wnt signaling pathway is downregulated and repressed in non-responder neurons. So, how do you test that? Just a quick idea of what a LEF1 is. LEF1 is called Lymphoid Enhancer binding factor 1. It belongs to a family of transcription activators and transcription factors. It does participate in Wnt signaling pathway and perform in its functions in complex with beta-catenin to activate transcription of downstream genes. Remember I said that during the Wnt signaling pathway, beta-catenin translocates into the nucleus and changes activation of downstream genes. No need to understand the pathway, but the idea being that when you having beta-catenin only is not enough, you need LEF1 to help with the transcription of those genes with beta-catenin. And that's an example here showing potential canonical binding site for Wnt called for the Wnt signaling called the Wnt response element, and it can be activated after binding of TCF-LEF and beta-catenin. So those are all working together. And here, just to remind you that LEF1 is downregulated, and over time it's the same thing. So that's just a plot showing the over time LEF1 in red here on NR is also non-responder is also downregulated. So we got this very complicated vector. How do you see beta-catenin activity in cells? So what we did is we took this very complicated vector, and I'm going to show you what in the right, what a summary of what it does. We got this from a company called Agene, and it's a very good company because it has a lot of constructs that you can purchase for not very much, and a lot of researchers actively deposit vectors there. So that is a DNA vector. So it's a DNA sequence that doesn't have to do with your genome DNA. It's a piece of DNA that is circular in the case because it's supposedly used for mammalian expression. So those vectors are the way of DNA and the ways in which we can add things to the cell that the cell doesn't have. So in summary, what this very complicated vector has that we wanted to use, if you look at the top, it has the 7 TCF-LEF1 binding sites. So those are the WRP sites where LEF1 and beta-catenin are going to bind and are going to promote transcription of downstream genes. And what do we have here as a downstream gene? We have a EGFP, which is an enhanced green fluorescent protein. And we also have an mCherry. So what this vector does is an indicator for us for two things. So if you look first in the GFP, it will tell us if the beta-catenin signaling is active, and if LEF1 is doing what it should be doing, GFP is going to glow green. And the second cassette here, it's an indicator that the cell got my construct, because I want to be able to see if I was able to put the construct inside the cell. Otherwise, I wouldn't know if something went wrong and the cells are not glowing green. But if the cells are, and mCherry is a red, fluorescent protein. So I'm looking here for the percentage of GFP positive cells on mCherry. So here's what does in the bottom, that's how things look in the culture. So in the left bottom panel, I'm seeing red cells, which means your cells are expressing, the cells got the construct and they are expressing cherry. So this promoted SV40 will express red constitutively. There is no restriction for expressing red. And then within the red cells, you can find some cells like this neuron on the top, or this other cell here, it looks like a progenitor wanting to be a neuron, that are now also expressing green. So what that means is these guys have beta-catenin activity. They have both the beta-catenin and the left one functioning, binding here and expressing the GFP. So that's our readout. So what I'm plotting here is, first I wanted to check if controls versus responders and non-responders had any problems transfecting them. Transfecting is adding the construct inside the cell and expressing red. And what I see is no. All the cells, and here's just because we had 3 controls, 3 bipolar responders, and 3 non-responders. So those are the actual individuals that we used. And here is a summary. So there is no significant changes between different cell lines with regards to expressing, to obtaining, to be able to be transfected with my construct. So that's good. Now, transfection efficiency is comparable. That's what we call. Now, when we look at the number of GFP positive cells, the green cells on over Sherry, so how many GFPs are, how many Sherrys are also GFPs? How many GFPs are on Sherrys? And when we plot that, we see differences. We see a decrease, a significant decrease in expression of GFP positive cells on non-responders compared to responders and control. And that indicates to us, or confirmed to us, that beta-catenin activation is significantly decreased in non-responders compared to control and responders. So we wanted to now connect our observation with changes in excitability of the cells. Remember, those cells, both bipolar responders and non-responders, are hyperactive. We have seen that before in collaboration with Shani Stan in the lab. So this is number of evoked action potentials. So those both responders and non-responders have more activity. We use a different system to look at that. We used a system called multi-electrode array, and multi-electrode array are plates that have embedded electrodes, and those electrodes can record extracellular voltage changes that are associated with electrical events. So this allows us to look at those, to look at detect neuronal activity simultaneously from many electrodes and many wells. So we can look at the dynamic network changes over time using the same population of neurons from different patients. And we can, the way the output of this analysis comes out are those traces or spikes. And then we can look at how many spikes we had. When you have a lot of spikes together, we have bursts, and how many bursts, and we can make some analysis out of that. And we can look at the frequency of the firing rate. And what I'm plotting here in red and blue, confirming that lithium responders and lithium non-responders at a specific time during their neuronal development have this hyperactive phenotype compared to control. And so we use that to confirm hyperexcitability. And what we wanted to see if we change left one expression, and then we use left one, we use control lines for that. If we decrease left one expression, same as it was happening to the bipolar, but on control cells, what happens to the neuronal activity? So we use another vector, which is a viral vector, and we made cell lines that I'm happy to talk about by email or afterwards if there are questions, but I'm not going to spend too much time. But the important point here is that we were able to decrease the amount of left one using a short hairpin vector on cells from controls. And then our question was, okay, so if I artificially decrease the LEF1, same as it would be happening to the bipolar patient in control cells, do we see any changes in neuronal activity? And that's our way to try to connect LEF1 deficiency to hyperactivity. So what we see here shown in this graph is, here is a plot, and here is just a graph over time. And here we're looking at day 20 and day 26 of differentiation. We see that the removal of LEF1 or decrease of LEF1 expression in control cells, and this is a scramble, is a control shRNA that we need to use. So when you compare the controls with a removal of LEF1, activity increases. So LEF1, decrease of LEF1 expression is correlated with increase in activity. So now it's a way that we're trying to find to connect LEF1 to hyperactivity. So decreasing LEF1 expression in control neurons is correlated with increased firing rates. So let me try to shift gears a little bit and trying to understand what else could be another drug that would be available to bipolar patients that have maybe somewhat similar, that are not as used, but it could have some beneficial beneficial effects on non-responders. And there's another drug called valproic acid, which sometimes given with lithium, sometimes without lithium, that acts through a different pathway. So VPA is a HDAC inhibitor, so it changes the way in which the chromatin is organized, but it also downstream controls transcriptional regulation. So we wanted to see if the use of valproic acid was going to change anything in terms of the Wnt signaling. So we used the same vector that I was talking to you before and looked at the GFP percentage compared to over in cherry expressing cells. And here I'm just looking at the non-responders. So as expected, treatment with lithium didn't do anything in terms of activity of beta-catenin, but treatment with VPA increased the number of GFP positive cells. So VPA was able in our system to increase at least a little bit, improve the beta-catenin signaling pathway by using that readout. So lithium treatment does not change Wnt pathway activity, but VPA increases the activity in non-responder neurons. And this is compared to control. So it's not a complete recover, but it does significantly increase the activity of that pathway. So, so we then wanted to see if a treatment of the cells went back to the RNA sequencing and looked at if treatment with VPA, lithium, or vehicle would change the neuronal the expression of the different genes in bipolar neurons compared to controls. And here I'm just showing a control experiment to show that bipolar and controls addition of lithium or VPA doesn't change the neurons that are made, so it doesn't change the proportion of neurons that we are sorting out. So that's just a control. And then we performed RNA sequencing on the sorted neurons. So as expected, lithium treatment did not significantly change gene expression compared to vehicle in non-responder neurons. However, so adding lithium to non-responder neurons didn't do anything in terms of anything significantly in terms of expression profile. So the neurons are exactly, the results are very comparative. However, adding VPA showed increase of 68 differentially expressed genes, so change the expression of 68 genes and at a significant level, which is this false discovery rate threshold of 0.05. That's just to make sure that we have a significance. So expression of LEF1 was only slightly increased, but there was a modest increase that this was not significant. But however, expression of non-beta-catenin gene targets, so the targets that supposed to change when you have a functional pathway. And I'm giving an example here of BMP4 and DKK1. Those pathways, those genes are upregulated significantly, suggesting that addition of VPA is changing in our non-responders is changing the beta-catenin activity. The other thing we wanted to do is we wanted to look at the differentially expressed genes between non-responders and controls that were recovered in non-responders after VPA treatment to a level that was compared to the controls. So let me pause quickly and explain that. And I have maybe 5 more slides. So I'm at the end of my presentation. I know I might be running a little bit over time. So the idea here is once you add VPA, some genes changed in non-responders. Which of those genes changed to the same level as control? So which are the genes that we call them like genes that are recovered? And when we look at the recovered genes, we do a plot to find out specifically what those genes are. And I'm highlighting them here in purple. Those are 63 genes. So those are the genes that became like control once you treated the non-responders with VPA. And those genes are in the category of glycoprotein, synaptic vesicle, transmembrane, and synaptosome. So a lot of genes were involved in voltage-gated channels. And here I'm just giving some of the examples. So the recovered genes are involved in synaptic activity. So our next natural question was, remember we saw those differences in activity. So that's the MEA, the mean firing rate, so activity of the neurons plotted. What happens if I add VPA? So adding VPA, when we will predict it would recover, as in decrease the hyper, calm down those neurons. And what we see here is that that's exactly what happened. So treatment with vehicle didn't keep them high, overly active. Treatment with lithium didn't change anything, and treatment with VPA calms the neurons down, decreases the activity of the neurons. So VPA, but not lithium, decreases spontaneous activity in non-responder neurons. I have a proposed, and then we have a proposed model for why lithium does not work on non-responders. And here is a summary of what we see. We see that increased VPA and increased LEF1, and we see that in increased beta-catenin pathway, so addition of VPA or presence of LEF1 promotes recovery of beta-catenin pathway, and that decreases hyperexcitability. So the idea is in our neurons, we have LEF1 deficiency. So even if you add lithium, even if you add lithium, the deficiency of LEF1 is still there. So even though you are increasing the levels of beta-catenin, you're not increasing the levels of its partner. So the transcription factor, the transcription expression that supposedly would help with hyperexcitability, it's not. So the neurons are still active because you don't have beta-catenin's partner. When you add VPA, you're changing levels of LEF1 binding partners, and you are helping with hyperexcitability. So lithium alone does not change LEF1 expression. So Wnt signaling, beta-catenin signaling, it's still downregulated, and the neurons cannot stop being hyperexcitable. Summary and conclusions from my talk: bipolar non-responder is transcriptionally distinct from bipolar responders and controls. Bipolar non-responder neurons have decreased canonical Wnt signaling pathway. So that's the pathway I was talking about. And we can see by using beta-catenin, TCF, LEF1 activity indicators, the green versus on top of the green over red. LEF1 expression is reduced in bipolar non-responder neurons. We show that. ectopically reducing LEF1, that means artificially reducing LEF1 in control neurons to make them more like bipolar neurons using a short hairpin RNA, induces hyperactivity. So we can somewhat simulate the hyperactive status just by decreasing the amount of LEF1. And finally, VPA treatment in non-responders recovers the expression of LEF1 targets and reduces hyperexcitability on the MEA, which is the multi-electrode array system that I showed that detects activity. So we believe, in conclusion, that modulation of LEF1 expression in neurons could be beneficial for non-responder patients. And we're working on studying the combined effect of both VPA and lithium. Idea is to pre-treat the cells with VPA to increase LEF1 and then treat with lithium to boost the Wnt signaling pathway. And we also want to test drugs that increase LEF1 activity or expression as a potential targets for bipolar NR. And there are some candidates out in the market already. And with that, I want to thank you for being here. And watching this presentation. I want to thank Rusty Gage and the Gage Lab, where most of this work has been developed. My very close collaborators and authors of this paper that was out last year, Renata Santos, Sarah Linker, Shani Stern. And we also have a collaboration with the NRCRC-RG Consortium and the psychiatrists, John Kelso and Martin Alda, for very graciously sharing patient cell lines with us. And I also would like to thank patients and families. I have some funding from Jensen and from National Institutes of Health. And I also want to thank these very bright women that work with me, Ariana, Ruth, Anna, Vipula, and Lynn, that were directly involved in this project. And without their hands, none of this would be possible. Thank you so much. And I'm happy to take questions.
Thank you so much for your presentation. I had a quick question. So in your research, you found that some genes were different in the non-responders and the control group. But when you first tested to see which genes were different, did you have any in mind that you thought were going to be different in the non-responders, or did you just find out after you tested for them?
We thought that GSK3 beta was going to be, so one of the, so we went for, because as I've shown before, the pathway is very intricate and there are a lot of players. So we went for players that were higher in the upstream in the pathway, and we didn't see any differences. So when we went for whole transcriptome sequencing, we were not always expecting to see, necessarily expecting to see changes in beta-catenin signaling, because we looked at some of the main upstream players and we didn't see differences. And then when LEF1 came as one of the top differentially regulated genes, we thought, oh, okay, we missed that. Which happens a lot in science. You think you know, many don't.
Mm-hmm. Very good, very good.
Thank you.
All right. So do we have other questions? It looks like Isabella has a question. You could unmute.
Can you hear me?
Yes. We are hearing you.
Okay. So my question is just, could this potentially also help treat epilepsy, since it's also involved in overactivity of neurons?
That's a great point. That's a great point. Some of the pathways, some of the channels of the calcium. So there are some. I quickly went through it because it's way too many details, and we just don't need to know all of that to get the gist of it. But some of the genes involved in calcium channels and I think we also have a chloride channel, have also been reported in some rare cases of epilepsy. So So that's a great point. We haven't even thought about that, but it's a good point that you raised.
Mhm.
Thank you.
Other questions?
I have a
hand.
question.
Oops.
Okay, go ahead.
So my question is, when I read the title of your presentation, the LEF1 gene, is it to do with the hyperexcitability or the lithium resistance, or both? Like, does it always express as both?
So let me see if I understood correctly. Let me know if I'm not addressing your question directly. The bipolar, and maybe I wasn't clear. The hyperactivity is always there on cells from bipolar patients, regardless of lithium responsiveness. But the only way one of the ways in which we can decrease the hyperexcitability with the responders is to add lithium. So that decreases the response. And the non-responders, we had no idea how to reduce their activity. When we saw changes in LEF1, we thought that maybe LEF1 could be involved in hyperexcitability changes. And that's why we tried to remove LEF1 in a control cell. And then we saw hyperactive neurons in a control cell that were not hyperactive before. That's not a direct link. We still don't know how LEF1 controls hyperactivity. And one way to do that would be to look at downstream regulators from LEF1. And we have some indications based on the VPA treatment too, that some downstream regulators of the pathway that is a Wnt signaling pathway and LEF1 is involved are genes that are regulating calcium channels and synaptic vesicles. So we think that LEF1 might be regulating activity via regulation of some of those synaptic vesicles and calcium channels, because changes in LEF1 can change downstream expression of those channels. But we don't know for sure because we haven't done the experiment yet. Did I get to your question?
Yes, thank you. You answered my question exactly and gave me more things to think about. I appreciate it.
Thank you.
Mm-hmm.
I see a raised hand from
Yes, Ryan.
Yeah, I got a
Dr.
question.
Hayes.
Yeah, thank you. Excellent talk. You just so much information. I teach instrumental analysis. We just did a fluorescence experiment. I saw the importance of fluorescence, data arrays, electrochemistry, great stuff. But in some of our research that we like to look at lithium, where you find lithium in nature. And I've always kind of had a question. How do we describe lithium's role nutritionally? Is your research enough to say, look, lithium is needed in the proper amounts to help regulate genes and their expressions? Can we say that? Do we know anything else where lithium, because like cobalt or some other transition metals, we can say, hey, those are needed because they're needed as part of certain vitamins and certain pathways. What can we say about lithium?
Yeah, you touched in the basis of many issues with the drugs that actually are being used in mental health, right? And lithium works when it works, but the research and the understanding of what exactly it's doing, and it's extremely poor. And for you to have an idea, it's not even clear how lithium goes into the cell. And there is also some evidence when you talk to clinicians that different patients uptake different amounts of neurons. You give the same amount and you find different amounts in different organs. Wow. And that so that's why also lithium can be very toxic to some people and not well tolerated by some people and not well tolerated out. So there's a lot of biometabolism that is absolutely not understood. And not that much is being done because it doesn't, well, we can go into a lot of discussion of the how pharma companies can push or not push research of their own interest, right? So I'm not going to go there. But that's a huge problem, right? Yeah.
That was helpful, just even to know what we don't know.
I don't know.
I think it's important for us to understand, and very interesting because I've seen studies where lithium levels in water, they seem to see incidences of less violent crimes. I don't know if you've seen some of. Yeah, there's a place in Texas and I think in Europe somewhere.
Yeah, lithium was being added to soft drinks at some point. Lithiated.
But it's toxic at certain levels. So there's this fine tuning effect there that if we understand its mechanism a little bit better, that would be interesting.
Just a follow up too on this idea that Ryan raised, but in a slightly different direction. Are there any studies that compares lithium carbonate with other lithium sources? Because I'm thinking, for example, like lithium combined with hydroxy acids like lithium lactate, lithium tartrate, because those are anions that are, naturally occurring, also non-toxic. They occur in different medicines as the counter ions. Do we know if there are any studies that compares these different sources?
That's a great point. I haven't really dig too deep in that in terms of different sources, but I think it's such an open field, and since we might not necessarily get too much interest in bigger pharma to understand those, I think that's something really somewhat low-hanging fruit in terms of basic research.
Right, exactly.
Right.
Exactly.
Look at different sources of lithium and how they affect cell lines or bioavailability.
So, for example, boron is a trace element that's needed in plants. I can't recall if it's needed in animals too, but perhaps if with enough studies, there might be some sort of indication that lithium could be sort of a trace element in terms of nutrition, again, along the lines of what Ryan was talking about.
Yeah, no, that's a great point.
Yeah.
Very not much studied.
Right. Why not lithium valproate?
Right.
Right. Right. That's good question, Ryan. Yeah. Do they use lithium valproate?
Or sodium valproate.
Yeah, or sodium, yeah.
Just combine the two, but
those would be two basic studies right there, right? You compare sodium valproate versus lithium valproate and see,
you wouldn't get the timing effect because that was part of your hypothesis, maybe.
Yes, and when you talk to clinicians too, they will say that. And again, big issue with many treatments for mental health. They will tell you that they start with lithium because that's what you always start. And then you go to the second line, it didn't work. And then by then, patient is either going through toxicity or going through not working at all. So it takes months and months, sometimes over a year to adjust. And then you go to the second drugs and you never really do it the other way. So why not try valproic acid first and then lithium? So, but I think they'll say too that sometimes the patient is on lithium and then they'll give a little bit of valproic acid just to see if it gets better. And then they take it out. But there's the studies are not very based yet. There are good studies on lithium responsiveness in terms of how much to give and when, and for how long. But even those are not necessarily directly linked with the chemistry of and bioavailability of the lithium in the body. Getting a little bit better on pharmacokinetics, but
yeah, I think I've read, and you've sort of like emphasized it in your research here, comparing VPA and lithium. I think I've read where they found that valproic is in fact better, and I guess with the nuance, maybe better for some than lithium. And so that's something that is really interesting, that again, hearing what you said today sort of reinforce what I've read in the past about the comparison between lithium and valproate.
yeah, I think there's so much to be done, especially that in this area, because there is maybe such an open chemistry Pandora box, because what they are doing mostly what pharma companies are doing is repurposing, but not rediscovering and not creating new compounds. So there's a lot of, because it's so expensive and you have to spend a lot of, you could make a lot of mistakes. But then what happens is the same compounds are being used for everything. And they are just a little bit better for one thing, or just the improvement is very minimal. And the clinicians will tell you they're very frustrated because they can't help their patients as much as they thought they want.
So maybe one more question, Gab, you want to unmute? Are you still there?
Yes. So my question was, it was based off Dr. Hayes's question. I was wondering, because I looked up that if there is like lithium in the body that's already there, and I found, I don't know if it's true, I found it in Google, but it says that there are trace amounts. So I was wondering if you think that like the variance in side effects when people are taking the anti-manic drug has to do with like maybe the interaction of the drug with the trace amount of lithium in the body already.
That's a good point. Yeah, I wouldn't know to respond for sure, but that's something interesting to think about.
Yeah, it looks like this is still a wide open field. I don't think there's a whole lot of research compared to other diseases and stuff and ailments. That's good.
The money.
Yeah.
The pharmaceutical companies want to follow, the big populations, and the money's not there, and that's too bad. For academia, I guess, can try to step in.
To step in. Well, I think this has been great. I had a self-interest in this topic because we are actually doing in collaboration with Dr. Marlene Murray over in biology. We are actually trying to create full spectrum bipolar disorder compounds. And so, yeah, we were very interested. And some of my research students were tuning in and all of that. So we may actually follow up with you offline and ask some more specific questions. Our area is based on valproate, in fact.
Interesting.
Yeah, and so we could have that discussion in some more detail later. But for now, we want to thank you so very much for this very informative, interesting topic.
Awesome. Thank you.
Yeah, that was great. It was very interesting.
Great discussions. Yeah.
Okay, thank you. Have a good evening.
You too. Bye.
Thank you.
Thank you.
All right, bye-bye.